There are many environmental agents which are not harmful, but they become active alkylating agents only after they are metabolized by the cell. So one such agent is a mixture of polycyclic aromatic hydrocarbons, commonly known as PAH. They are formed by the incomplete combustion of the burning wood or coal used as a fuel. Similar type of polycyclic aromatic hydrocarbons are also present in tobacco smoke and charbroiled meats. There are more than 100 different types of PAH compounds present in the environment. So here are some examples. So these include benzopyrene. These two are benzopyrene, and this is chrysin, and this is dibenzanthracene. So here are some examples of polycyclic aromatic hydrocarbons which can damage DNA. The common structural feature in all polycyclic aromatic hydrocarbons is the presence of two or more fused aromatic rings. These are not able to damage DNA unless they are metabolically activated in the cell. So they may get entry into the cell from the environment, and into the cell they are activated, and these activated products can damage DNA. Otherwise, these PAH are not harmful for the DNA. One of the polycyclic aromatic hydrocarbon is benzoapyrene. It is converted into an active epoxide alkylating agent through a metabolic pathway in the cell. So here is the pathway which converts this pH into epoxide, which is a damaging agent. So this is benzoapyrene. This is activated by the oxygen, and it is converted into benzoapyrene 7,8 epoxide. So this here you can see this is 7,8 epoxide, and in the further pathway water is added, and this epoxide 7,8 epoxide is converted into benzoapyrene 7,8 dihydrodiol. So again, this is not damaging for the DNA, and this is again activated by the oxygen, and it is converted into the highly reactive or damaging agent, which is benzoapyrene 7,8 dihydrodiol 9,10 epoxide. So this epoxide will alkylate DNA. So cytochrome P450 enzymes are involved in the activation of this PAH. These enzymes are not very specific. So if they are not very specific, they may act on some other substrates. So in the same way, they act on pH such as benzoapyrene, and add oxygen atoms to form reactive three-membered epoxide rings. These epoxides then alkylate DNA, causing replication errors that result in mutations. And due to these mutations, the cells ultimately convert, the normal cells ultimately will convert into the cancer cells. So here are some examples of the damage, DNA damage by pH. For example, so this is pH, this one, and here it is attached with the C8 of guanine, and in this way it forms a monoadduct. Similarly, here this hole is attached with the N7 of guanine. This is N7 atom of guanine. So in these two ways it can damage guanine base, but it can also form monoadduct with adenine. So here the example of monoadduct of adenine, and here it is attached with the N9 of adenine. So in this way, these monoadducts are formed by the active agents which are formed after metabolic activation of PAH.